Alright guys, gals, let's get started with our lecture discussion on DNA and cellular division. We're going to take this presentation and break it up into two segments. We'll have one segment on how to replicate DNA, and then we'll have another segment on cellular division with a heavy emphasis on mitosis. So first, before we talk about these processes, there's a couple structures in the cell that we just have to cover. And I know we haven't had our discussion on structures of the cell and organelles of the cell, and we're not going to cover all of them right now. We're just going to cover the bare minimum that we need to get over DNA and cellular division. The first thing we need to cover, the first thing we need to mention is the cell membrane. The cell membrane is going to be on the surface of the cell. It is going to separate the interior environment of the cell from the exterior environment of the cell, or also known as the intracellular environment from the extracellular environment. And there's a key structure on the intracellular environment, on the inside of the cell. It's the largest structure on the inside of most cells, and that's going to be the nucleus. And in that nucleus, what we really want to emphasize right now is that we store DNA. We just store that genetic code inside of the nucleus of the cell. And in the nucleus of the cell, there is a specific way that we store DNA. It's not like my sock drawer where I just throw everything in there randomly and let it become a giant disorganized mess. There's organization. There's a rhyme and a reason to the chaos on the inside of the nucleus of the cell. And we take all of that DNA, the centimeters upon centimeters of DNA, and really that's a really long piece of a microscopic molecule, and we're going to wrap it around little pieces of protein called histones. And those histones you can think of like spools that you wrap thread around. So we take the DNA, we wrap it around the histones, and then the histones are coiled around themselves, and there's a special name as we keep wrapping and wrapping and wrapping and wrapping. As we keep wrapping and supercoiling the DNA, eventually we end up with a big piece of DNA that's large enough for us to see with a regular visible light microscope. And this piece of tightly coiled DNA is called a chromosome. If it was loosely coiled DNA, we'd call it chromatin, which is another name for these proteins that the DNA is wrapped around and the DNA itself. But tightly coiled DNA is called a chromosome. And there's a very special stage of cellular reproduction where the DNA is coiled in the chromosomes and there's these little branches, or the chromosome kind of makes an X. And while that chromosome is making the X, each branch of that X is called a sister chromatid. So here we have the P arm right here of this one chromosome, which is identical to the other P arm of this chromosome. So these are two sister chromatids, the short arms, and then down here we have two more sister chromatids. We're not going to spend time, this is not a genetics class, so we're not going to focus on P arms versus Q arms versus telomeres. Right now, really what I want to emphasize to you is that these sister chromatids are genetically identical within this chromosome. There's another key structure on the inside of the cell. This is going to be inside of the cell but outside of the nucleus. And this key organelle or structure is called a centrosome. I've always thought that a centrosome looks a lot like a churro, or like, that cinnamon sugar Hispanic dessert. I mean, they're delicious. And when we look at the centrosome, it's going to make a lot of projections coming off of it. These projections are called microtubules, and they're part of the cytoskeleton. We need these microtubules from the centrosome during our discussion of cellular reproduction, because these microtubules help to organize the chromosomes as the chromosomes are being pulled apart and moved throughout the cell. So we have some essential cellular structures with the cell membrane, the nucleus, the chromosome, and the centrosomes. So let's talk about DNA replication. Before a cell divides, it needs to duplicate its DNA. And this is a very key function, because if it didn't duplicate its DNA before it divided, those daughter cells that are produced, wouldn't have the complete set of instructions. And within a couple rounds of cellular division, we would cease to exist as a species. And certainly you would cease to exist as an organism. So this ability to duplicate the DNA before we duplicate the cell is essential. Within the DNA, we have genes. And a short definition of a gene right now is just going to be that a gene is a segment of DNA within a chromosome that gives a specific set of instructions. So, as we look at this DNA, the DNA is made of genes. Lots of genes strung together make a chromosome. The genes themselves are the individual sets of instructions within the DNA. And with these genes, the DNA in the nucleus controls the cell. And because of this process, we have to replicate the DNA very precisely. It's kind of like when you copy or duplicate a file on a computer. If you duplicate the file and you don't duplicate it correctly, oftentimes the file just doesn't work at all. And the same is true of a gene. Many instances of gene replication result in the gene not working if it's not copied correctly. As we're copying our DNA, there's a key vocab term or a key idea here. And this is the idea that one nucleotide will always pair, or almost always pair, with another nucleotide. In this case, adenine will pair with thymine. A pairs with T, typically with 2 hydrogen bonds in between. And then cytosine pairs with guanine, so C to G. And this typically will have 3 hydrogen bonds binding them together. So if you know the sequence of one strand of DNA, maybe it's AAGC, then you can know the sequence of the other strand of DNA, just based off of this law of complementary base pairing. So as we're going through DNA replication, there are some key steps. To summarize all these key steps, we need to take the DNA apart, read the information, copy the information, and zip the DNA back together. So as we're looking at this DNA, step 1, we need to unwind that DNA from the histones. Let's back up a couple slides here. Here are some histones that the DNA is wrapped around. Before we can process that, Before we can process that DNA, we need to unwind the DNA from the histone. Step 2 is one of those enzymes that's the butt of a lot of jokes, or the punchline from a lot of jokes, and that's DNA helicase. DNA helicase is an enzyme. Let's look at the name here. It ends with ASE. That's a big clue that this is an enzyme. What does this enzyme do? It does something to the DNA helix, hence its name, DNA helicase. Specifically, DNA helicase unzips the double helix of the DNA. It breaks apart those hydrogen bonds that holds the A and T and the C and G together. So as we unzip the DNA with DNA helicase, we're exposing the information in the genetic code to be read. The point that DNA helicase unzips the DNA is known as the replication fork. And we can see that down here. So here we have DNA helicase. It's unzipping the genetic code. It goes from double-stranded to single-stranded. And this point where helicase is taking a single strand and branching it into two, or double-stranded DNA and branching into 2 single strands, is known as the replication fork. Step number 3 gets to be a little bit more complicated. During step number 3, we're going to take a new enzyme called DNA polymerase, so polymerase, for those of you with a Midwest accent. The ASE, again, is a clue that this is an enzyme. What does this enzyme do? It does something to the DNA polymer. Recall back to our discussion on macromolecules. A polymer is a large molecule made of many subunits. Poly meaning many, mer meaning subunits. So a polymer is something made of many subunits. This particular enzyme works with something made of many subunits of DNA, or a big piece of DNA, in other words. So DNA polymerase is going to make new macromolecules, new polymers of DNA, or new strands of DNA. DNA polymerase will read the exposed bases that have been exposed by DNA helicase. And we have 2 DNA polymerases, one for each single-stranded piece of DNA. And because DNA is directional, just like you and I read the English language from left and right, there's a direction or an orientation that we need to read the DNA at. So one DNA polymerase can just add continuously. The other DNA polymerase needs to add in segments. So notice how this DNA polymerase is going from right to left, adding continuously. And then down here, we're going to have DNA polymerases going from left to right. So this DNA polymerase is traveling in an opposite direction compared to the DNA polymerase on the top of the screen, which is traveling to the left. We're not going to spend a lot of time talking about Okazaki fragments or the sliding trombone model. This is not molecular biology. This is human anatomy physiology. So we're just going to leave it at the DNA polymerases need to synthesize or add DNA in opposite directions. The end product of the thing that we really care about is that we went from one strand of DNA at the beginning, one parent strand, to two fully identical daughter strands of DNA. Now, as we're having this DNA polymerase synthesized, this continuous piece of DNA that's being synthesized on top, this is the simple one, because it's just, let's just make it bigger and bigger and bigger. Down here on the bottom of the screen, where we have to synthesize the DNA in segments, we're going to need to link those segments of DNA together. And there's a special enzyme called DNA ligase, shown here as the blue donut. And this DNA ligase is going to take those individual segments of DNA that are being synthesized as individual fragments, and it will link them together. It will ligate them together. And again, at the very end, step E here that we're going to emphasize, we go from one double-stranded piece of DNA to two double-stranded pieces of DNA. A key concept or idea during DNA replication is semi-conservative replication. If we look at these parent strands, we can call these the original strands of DNA. We'll call those the original or old strands because they're parental, so they're old, right? So the parental strands of DNA are used to make the daughter strands or the new pieces of DNA. And as we are looking at this here, we go from blue strands of DNA, and notice how this new strand over here, this new double helix over here, is half blue and it's half orange. The blue backbone represents the old parental DNA, and then the orange backbone represents the new daughter DNA. So when we think of semi-conservative replication, these new strands of DNA are half brand new and then half recycled from the parent strand. It's semi-conserved with the new helix. And then finally, step number 4. This process of replicating DNA is going to be multiplied many thousands of times over per chromosome, per strand of DNA. And we're going to need to take this new chunk of DNA and package it up so it can be processed. So we'll take all of these pieces of DNA and repackage them. And to give you an idea of the enormity of this job, in every cell, or every somatic cell or regular cell of the human body, pretty much everything but a sperm and egg cell, we're going to have 46 chromosomes. Yes, there are a few exceptions. Red blood cells are a notable exception, but generally speaking, if it's a regular cell of the human body, it's going to have 46 chromosomes in it. And we can replicate all those chromosomes in the cell of the body in about 6 to 8 hours. And we're talking about thousands upon thousands of polymerases working all at the same time with millions upon millions of histones being unwound and rewound. It's a highly complicated process that goes off almost seamlessly. Every once in a while there's little mistakes, but generally speaking, there are very few mistakes. But there are some mistakes that happen every now and again. No enzyme is perfect, and every once in a while, DNA polymerase is going to have a replication error. Instead of adding a T, it'll add a G, or something of that nature. So what we have right now in our DNA replication process are some proofreading mechanisms. And as we look at these proofreading mechanisms, they're going to find a way for us to double check the new strand of DNA, that daughter strand of DNA, and compare it to the parent strand of DNA. And this is a highly reliable system for double checking. And ultimately, when we look at the final mutation rate or the final rate of error, we're looking at about 1 base pair mistake for every 1 billion bases replicated. So this DNA polymerase is one of the single most accurate ways to replicate information known to humanity. It's by far and above more accurate than human beings, and it's also more accurate than most computer systems that are in place to replicate computer files when we think of the bits, the zeros and the ones within computer files. If we do have that 1 error per million bases, there'll be a mutation that occurs. And this mutation that occurs is just a mistake in DNA replication. Some things that contribute to mutations include radiation or viruses or chemicals. Sometimes it's just random dumb luck. Some mutations don't do anything at all, depending on where the mutation is within the chromosome or how the mutation manifests itself. And we'll talk more about this with protein synthesis. Some mutations are silent mutations because ultimately they don't affect the amino acids that go into the protein. Some mutations, though, are going to cause the cell to self-destruct. Some can cause the cell to reproduce out of control, in other words, to become cancerous. Or some mutations, particularly mutations to the germline, to the mutations in the ovaries and or testes, can cause genetic defects in future generations. And these are the scary mutations, mutations to the sperm or egg, because those carry from generation to generation. I would play this video for you right now in this recording, but I've done this in the past and I know that YouTube doesn't like playing other YouTube videos within a YouTube video, so due to copyright issues, I'm going to tell you to click on this link because I don't want YouTube to pull my video and my lecture recording. But let's just make sure quick that the link works. Yes, the link works. Okay.
Soccer play.
So make sure that you download this PowerPoint, click on the link. This is a fabulous video to watch. It will really help emphasize DNA replication for you. And that's all we have for our recording on DNA replication. If you have any questions, please be sure to shoot me an email, come to my office hours, or ask the supplemental instructor. Happy studies!